So today we're going to talk about when a single gene has more than one allele. And so we've already sort of alluded to that in some ways. So if we have gene A, we might have the dominant allele A and the recessive allele A. And so let's say that the dominant allele A confers red color, but the little a only confers pink color. And maybe we have an A1 allele that's still for gene A, but it's a different version, and it gives us sort of magenta colored flowers. And maybe we'll have an A2 allele that gives us orange colored flowers. So we've got multiple alleles, but they're all different versions of the same gene, this red color gene. Now, if I were to discover a new mutant, I have a new mutant over here, a new mutant flower, and it's pink. And I want to ask, have I discovered a new allele of gene A? Or is this a whole separate gene that affects flower color? So I'm wanting to ask if I have another allele of gene A or a separate gene, a mutation in a separate gene that affects flower color. And so we're going to work through a problem that lets us solve this question, okay? So I'm going to erase our original alleles here. We have a new mutant that is pink, okay? And in order to answer the question whether we have a new allele of gene A or a whole separate gene that affects flower color, we're going to perform a complementation test, okay? And in this case, we're going to take our new mutant, the pink mutant, and we're going to cross it together with, let's remember that our gene A has the dominant allele red and the lowercase allele A, which gives us only pink color. So we're going to cross our new pink mutant here together with our old pink mutant. And what do you expect to come out of that cross? Well, you would expect, right, that these flowers are going to be pink, okay? And that would make sense. Let's say here's our old genotype A, and if we have another allele in gene A, let's call it little a prime, okay? Then this mutant, or this heterozygote is going to be little a prime. It's still heterozygous because it got a different version of the mutation from each parent, but the consequence is it has no functional versions of A. So here we would say that there is no complementation, and we'll talk more about that in a minute. No complementation, and we still see these original phenotypes, okay? Now, on the other hand, if we cross together our new mutant that's pink and our old little a mutant that's pink, and we got red offspring, that would be pretty surprising. Now we know that this red offspring has to be heterozygous, okay? In this case, we'll say we have two little a's of this, but we're going to say there's a separate gene B for which this mutant, this original gene A mutant, still has functioning copies, okay? And now this new pink mutant that we've identified has functioning copies of gene A, but it has a mutation in a whole new gene, gene B, okay? And then this cross here, our F1, must be heterozygous. So we'll get a big A from one parent and a little a from the other, a big B from one parent and a little b from the other. And so this plant turns out to be red because it's got a functional version of A, and it's got a functional version of a second gene, B, which affects flower color here. So in this case, we would say there's complementation. And what the complementation means is, in this plant, It's pink because it doesn't have a functioning version of big A. This plant is not pink. Its little a's are complemented by the presence of a big A from the other parent. And likewise, this plant is not red because it doesn't have a functioning version of B, but it's complemented in the heterozygote by the big B that it received from the other parent. So we have complementation here, which restores the wild type phenotype. Okay, and this suggests that the two mutations are in two genes. Complementation means two genes are involved. Over here, where we had no complementation, we were able to conclude that the old mutation and the new mutation don't restore the wild type phenotype. So they must be mutations that are occurring in the same gene. So one gene, two mutations, one gene. Over here we have two mutations, but two genes. So that's what a complementation test will let us know. So where on earth would we ever use a complementation test like this? I'm going to erase these details, and we're going to talk about a different scenario. Okay. We're going to talk about a different scenario where you are working in a lab. You're working in a fruit fly lab, and you have decided to study fruit flies who normally have red eyes, but you're looking at mutants that have white eyes. Now, as you prepare this study, you've got your wild type flies. They have red eyes. Okay. And then you receive three mutants from different labs. You receive a redless mutant, which we're going to call RD. Okay. You receive from another lab a mutant called not red, which we're going to shorten to NT. And then from a third lab, you receive a fly who has a mutation called white, which we're just going to call W. Now, these are three mutants that all have the phenotype of not having red eyes anymore, unlike the wild type flies. You've got three mutants with white eyes. And you, as you start your project, you want to ask, now, are these three mutants all in the same gene, but with different names? Or are they three different mutations that all affect fly eye color? And we'll use a complementation test to try and figure that out. So in order to do this, so I'm going to write down here, do we have multiple alleles or multiple genes? Okay. So what I want you to do is perform a series of crosses together. So we'll cross, for example, redless times not red, NT, and figure out what that progeny looks like. Redless times white, what's the phenotype of that progeny? And then not red times white. And actually, I can give you the data already over here. When we cross redless times redless, we have no complementation. When we cross redless times not red, we do have complementation. And when we cross redless times white, we don't have complementation. Okay? Next, we're going to add in not red. So when we cross not red and not red together, we don't have complementation. That's good. That's a good conclusion because these should be the same exact gene, so they don't have complementation. And when we cross not red with white, we do have complementation. And then last but not least, when we cross white flies with white flies, we don't have complementation. Okay? So this is our data. And from this data, we want to figure out, do we have multiple alleles that affect red eye color, or do we have multiple genes that are affecting red eye color? So like I said before, the fact that when we cross redless times redless and we have no complementation, that's a good thing. We know that the redless mutation then is just in one gene. It's not something weird going on. And the same thing when we cross not red times not red right here. We don't have complementation, so we have no complementation when we do redless times redless, when we do not red times not red, and when we do white times white. That's good. Each of these are single mutations that are segregating as we would expect. We also see no complementation when we cross redless times white. And is that the only other time? And when we cross, oh, no, not red times not, that's the only other time we have no complementation. And we do have complementation when we see redless times not red, and not red times white. So when we cross flies with the not red phenotype times the white phenotype. They're the same phenotype, they just have different names. So we don't have complementation in this case. Makes sense, makes sense, makes sense. And remember that no complementation means one gene with two different alleles. Okay. Complementation, then, we have two alleles, but they're in two different genes. Okay. So what this means is that RD, redless and white, must be alleles of the same gene. Okay, these have the same gene. While redless and not red are in two different genes, and not red and white are in two different genes. If we were to draw this on the chromosome now, here's our chromosome, we could say we have the not red gene over here, and then we have one locus where redless and white are both located. They're actually the same gene. We've just, remember we collected this from one lab and this from a second lab. These are actually mutations in the same gene. So these are alleles of the same gene, whereas not red is a mutation in a completely separate gene. Okay? In this case, because we're talking about red versus white eyes, no complementation is going to show us white eyes. And then complementation is going to restore that original phenotype of red eyes. Okay, just in case you're wondering what these complementation tests look like. So, as a take-home message here, complementation testing, our favorite, lets us tell whether two or more mutations are in the same gene or whether they're in separate genes. Are these mutations multiple alleles of one gene, or are they representing multiple genes affecting a single phenotype? So I hope this helps you to understand a little bit more about complementation testing, and we'll be doing some practice problems about complementation testing in class and on your homework. Thank you.